Our protocol provides a simple, yet robust approach to identify and characterize competitive mechanisms between different strains of Vibrio fischeri bacteria. This method uses stable plasmids that encode different antibiotic resistance genes and fluorescent proteins that allow for differential selection and visual discrimination of each bacterial strain in a mixed culture.
To begin, use a sterile toothpick to scrape the cells from the agar plate in the size of a grain of rice. Resuspend the cells in a 1.5 milliliter microcentrifuge tube containing 1 milliliter of LBS broth. To break up the cell clumps, press them into the side of the tube and then pipette up and down vigorously. Close the tube in vortex for 1 to 2 seconds and repeat if the cell clumps are still visible until the sample is visually uniform. Repeat these preparation steps from cell scraping till vortexing for all samples. Measure and record the optical density at 600 nanometers for all samples. To obtain an accurate OD measurement, dilute the samples up to 10-fold using LBS broth. Then normalize each sample to the desired OD 600. To mix the reference strain and competitor strain in a 1-to-1 ratio, add 100 microliters of each strain normalized to OD 1.0 to a labeled 1.5 milliliter centrifuge tube, and vortex for 1 to 2 seconds. As a control, mix the reference strain with a differentially tagged version of itself, and vortex as well.
It is critical to achieve the correct starting ratio, as it can considerably change the outcome of the experiment. To ensure success, this step may require optimization for a given bacterial species.
After repeating this step for each biological replication,